What's up, biology students? In our last video, we talked a lot about chromosomes and the role they play in inheritance of traits. In this video, we're going to talk about mitosis and the cell cycle, the process by which the cell divides and sorts those chromosomes and passes them on to the next generation of cells. Cells are the basic unit of life, and like all life forms, cells grow, develop, and reproduce over their lifetime. This diagram represents the steps in the cell cycle, which you can think of as kind of like the life story of one particular cell. When a cell is resting, which means it's functioning but not really growing or developing, we would call that phase G0, and some cells stay in this phase for a really long time, while others rarely, if ever, enter this phase. When a cell is developing in preparation to divide, it enters a period of growth called the G1 phase. During the G1 phase, most of the contents of the cell, with the exception of the cell's DNA, are copied and replicated, and the cells increase in size. Then the cell enters the synthesis, or S phase of the cell cycle. To synthesize means to build, and this process is the phase when cells synthesize or build new DNA by way of DNA replication. After this phase of the process is complete, the cell enters another period of growth called the G2 phase. During G2, the cell double-checks its work, making sure that all the chromosomes inside the cell were duplicated accurately, and making any repairs that may be needed. All three of these phases, G1, S, and G2, are lumped together as part of what is called interphase, the part of the cell cycle in which the cell is not actively dividing, but in which the cell is preparing to divide. As you can see, interphase makes up the majority of the cell cycle in terms of the time taken to complete this phase. After interphase, when the cell has completed the G1, S, and G2 phases and is ready to divide, the cell begins a process called mitosis. The word mitosis is often used interchangeably with cell division, but that's not technically correct. Mitosis really only refers to the division of the cell's nucleus and the process by which the genetic contents of the nucleus are divided into two equal sets that are identical to the original set of genetic material in the original nucleus. There are a number of organisms whose cells do go through mitosis without necessarily dividing into two new cells. Fungi do this all the time, actually, and the result is one cell with two nuclei, rather than two new cells, each with one nuclei. The term cytokinesis refers to the division of the cytoplasm, which splits the one cell into two new daughter cells. So both mitosis and cytokinesis need to occur in order for a cell to divide, and to ensure that each new cell has a full and complete set of chromosomes. Often, cytokinesis and mitosis overlap with one another, but they are different processes. Basically, mitosis divides up the cell's genetic material into two new nuclei, and cytokinesis packages these two new nuclei into two new cells. Most of the time, the cell's DNA is loosely organized into a mass of DNA and histone proteins called chromatin. Human DNA contains approximately 3.5 billion base pairs, and it would be a nightmare to try to sort out all of that DNA into two separate nuclei if it was just a tangled mass. And therein lies the advantage of organizing our DNA into chromosomes. For humans, our DNA is organized into 23 pairs of homologous chromosomes, or 46 total chromosomes per cell. Following DNA replication, each of these 46 chromosomes contains two identical sister chromatids, which we can see here in this figure. During mitosis, these sister chromatids are pulled apart in opposite directions, and each is packaged into one of the two new nuclei being formed, ensuring that each new cell gets the right set of genes. Although this process of growth, DNA replication, sorting, and repackaging is a continuous process, we give names to certain points along the way, certain important benchmarks to help us figure out where we are and what should be happening during different phases of mitosis and cytokinesis. Interphase, you may recall, is the period in which the cell grows, makes copies of its organelles and its DNA, and generally prepares for division. Interphase is also the longest phase of the cell cycle by far, which isn't really clear from this particular figure. Prophase is the first phase of mitosis, followed by metaphase, anaphase, then telophase and cytokinesis, which often occur at the same time during the cell cycle. After cytokinesis, the two new cells, called daughter cells, either enter a period of rest called G0 or re-enter the cell cycle by transitioning to the growth or G1 phase of interphase. Let's take a look at each one of these phases, starting with prophase, the first phase of mitosis. During prophase, duplicated chromosomes begin to condense from the loose mass of chromatin found in the nucleus during interphase. We can see here in this microscope photo what that looks like in real-life cells. Meanwhile, these structures called centrioles, which are present in animal cells and in certain lineages of plant cells, migrate to opposite ends of the cell. These centrioles will help regulate the movement of chromosomes later on. As the centrioles migrate, structures called spindles begin to form. Near the end of prophase, the nuclear envelope breaks down and the nucleolus disappears. Chromosomes begin to move toward the center of the cell. The next phase of mitosis is called metaphase, and metaphase marks the point in the process in which homologous chromosomes line up in the center of the cell, which we can see here in this diagram, as well as in this microscope photo here. The spindle fibers that began to form during prophase, and whose formation was regulated by the centrioles we talked about earlier, connect to each chromosome at the centromere. The next phase in mitosis is called anaphase, and this is when the spindle fibers pull apart the centromeres of each chromosome, separating the sister chromatids and pulling them towards opposite ends of the cell. In this microscope photo, we can see what that looks like, both early and late in the process, as the chromosomes are being pulled farther and farther apart. At this point, we enter telophase, the last phase of mitosis. During telophase, chromosomes separate into two distinct groups near the spindle apparatus at the poles of the cell. The nuclear envelope begins to reform around each group of chromosomes, forming two new nuclei at either end of the cell. Finally, the spindle apparatus begins to break apart. Chromosomes begin to unwind into a loose mass of chromatin, and a new nucleolus becomes visible in each new nuclei. Meanwhile, the process of cytokinesis begins to divide the cytoplasm of the cell. This happens a little differently in plants and animals. Here, at the top, we can see how cytokinesis happens in animal cells. After anaphase, microfilaments form around the center of the cell. Then, microfilaments constrict, forming a cleavage furrow, pinching inwards from the outside of the cell. Eventually, the cell is completely pinched by the microfilaments and two new cells form. In plants, it happens a bit differently, because these plants also have to build a new cell wall in addition to dividing the cell membrane in two. In plants, after anaphase, carbohydrate-rich vesicles form around the center of the cell. These vesicles fuse, and a structure called a cell plate begins to form. This cell plate grows from the center of the cell outwards toward the edge, and once the cell plate is completely formed, two separate cells will have been created. At this point, whether we're talking about plants or animals, the cell cycle has been completed and is ready to begin again. The timing and events of the cell cycle are regulated by a group of proteins called cyclins. In this graph, you can see how the concentration of these cyclins changes throughout the cell cycle. Since cyclins are proteins, that means the instructions for building them are encoded in our genes. And when these genes are expressed, cyclin proteins are built, and the concentration of these cyclins rises in the cell. When these genes stop being expressed, protein synthesis stops, and the concentration of cyclins begins to drop. For example, we see that the concentration of cyclin E spikes right when the cell is transitioning from G1 phase to S phase, and we might therefore infer that cyclin E has something to do with beginning the process of DNA replication that occurs during S phase. Likewise, we might infer that cyclin B, which spikes right at the end of G2 phase of the cell cycle, has something to do with beginning the process of mitosis. This is just another example of the many important roles that protein molecules play inside of our cells. The cell cycle in prokaryotes is a little different. For starters, since prokaryotes have no membrane-bound organelles, and therefore have no nucleus, that means that prokaryotes don't go through mitosis. Prokaryotes do contain chromosomes, though, just smaller, simpler chromosomes, and these need to be replicated, just like the DNA in a eukaryotic cell does. After DNA replication, the cell wall and membrane of this prokaryote begins to divide until a cross wall forms completely around the divided DNA. The two new cells separate, and the process begins again. In prokaryotes, this process is called binary fission, literally the splitting of 1 into 2. And we can see a snapshot of this process here in this electron micrograph of a bacterium. All cells come from other cells. This is one of the basic tenets of cell theory, and the process of cell division is what makes that possible. And as long as every step in the cell cycle goes correctly, from DNA replication during S phase to the separation of chromosomes during mitosis, then each new cell will have the genetic material and important structures necessary to continue living and functioning. In multicellular organisms like us, this process leads to the formation of many trillions of cells that make up our bodies, and continues to happen every day of our lives to replace the cells that eventually wear out and die, keeping our bodies healthy and helping us to heal from injuries and disease. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand the cell cycle, mitosis, and cytokinesis, and how these processes help cells to divide and reproduce.